the DNA, and in this fragment, this adapter is ligated here. Okay, so as the adapter is ligated here, so machine is only able to read this particular sequence from the forward side, and that is why here for the single line sequencing you are getting only a one output file, because the adapter is ligated at one end. So it can be from forward, or if the adapter is ligated at from reverse, so machine is going to read this particular sequence from the reverse side. So this is what about the single end sequencing. And for the parent sequencing, actually for the parent sequencing, adapters are ligated at both the end. So that is why machine is able to read the fragment from both the side. Okay, so here in this parent sequencing, it is able to read the fragment from both side of the from a both side, like forward as well as reverse. So if you will see this diagram here, the adapter is ligated at both the ends. So machine is reading this particular fragment of DNA from the forward side as well as from the reverse side, which means from the 5' end and from the 3' end. So that is why here in the parent sequencing, we are getting two output files. That is one for forward, another for reverse. Means in the forward file, you will get all the sequence or all the reads which is read it from the forward side, and in the next file, you are getting all the sequence which read it from the reverse side. So actually, it's totally up to you that you wanted to go with the single end sequencing or with the paired end sequencing. But the parent sequencing will give you more efficient result because the one fragment is read from both sides, the confidence is much higher, right? But again, the choice is up to you because if it's a parent sequencing, cost of the sequencing will also increase. And one more thing, whenever we are aligning the single end sequencing or a paired end sequencing data to the reference genome, because as we have already discussed about the DNA sequencing, where we need to be align our reads to the reference genome. So we are getting the alignment file after that. So whether it's a single end or a paired end, you are getting only a one alignment file, only a one output file, because whenever we are aligning the reads to the reference, it is going to align at the same position, doesn't matter what is the direction of that, right? So that is why here we are getting only a one alignment file. So this is what actually about the types of sequencing. After that, the one more concept which is very important regarding to the NGS, that is a Fred quality score. So the Fred quality score, the name itself, it is suggesting that it's a quality score. But quality of what? So it's a base quality score. And one thing I just wanted to clear here before starting the Fred quality score, that it is assigned to each base. Okay? It is assigned to each base. Means this is a quality score of the base. So as we all know that the machine is detecting the bases, machine is predicting the bases. So base which is detected, or you can say a predicted by the machine, has some quality score which tells you about the accuracy, that how much accurately that base was called. Means basically here you can say in the Fred quality score you are getting the base call accuracy. And as it is telling you about the accuracy, there should be some cutoff. At what cutoff we must say that the base is good quality or not? So the standard cutoff for the Fred quality score, it should be greater than or equals to 30. Means if the base is having the Fred quality score greater than 30, it's a good quality, and if it is having less than 30, that's not a good quality base. This is a standard cutoff, but according to your data, if you wanted to increase, if you wanted to decrease, that choice is totally up to you. But now, one more question arising over here, that why we decided to take this cutoff as a 30? How much percentage that particular base is accurate if the Fred quality score is greater than 30? So for that, here, if you will see this table, like here we are having the Fred quality score cutoff 10. So it means a machine called that base 10 times. Out of that, 9 times it called correctly and 1 times incorrectly, which means the accuracy of that base is 90%. Because if here in this first two column we are having the Fred quality score, in the second column you are having the probability of incorrect base call, means how many times the base is incorrectly called and the percentage of accuracy. So here you are having the 20 Fred quality score, which means the machine called that base 100 times, but out of that, 99 times it called correctly and 1 times incorrectly. That time, the accuracy of the base is what? 99%. Now let's talk about our standard cutoff 30. So it means machine called that base 1000 times. Out of that, 1 time it is called incorrectly, which means a 99.9 times it called correctly, and that's actually the good cutoff. That is why we have decided to take the cutoff 30. So greater than 30, you can see the accuracy is also increasing. When the Fred quality score will increase, the accuracy will also increase. But one thing, if you will notice here, there is a no Fred quality score which is giving you a 100% cutoff, right? Because there is a no machine which is able to read the 100% accurate base. So this is about the Fred quality score. Now, as actually whenever we are getting the read sequence, along with that only we are getting the Fred quality score. But in which format and all we are getting, that I will discuss you during the file formats of the raw data. But for now, you just understand this Fred quality score is the quality of the base which is called. So after the Fred quality score, the next term actually I wanted to discuss over here is mapping quality. So what is a mapping quality? Now, base quality is a quality of a base, and mapping quality is a quality of mapped read. Because here in the DNA sequencing or in whatever the NGS application you have taken, in that we are aligning, maybe if you are aligning the reads to the reference genome, at that time you are getting the cutoff for the mapping quality, which is telling how read is aligned to the reference genome. Okay, so in the mapping quality, it tells how the read is aligned to your reference genome. Means, let us consider now. So consider, just for example, like this is your reference. And here we are aligning our reads to this reference genome, right? So let us consider, like, as the reference genome is very huge as compared to the read sequence. So one read can align at a multiple position. So maybe let's consider this is a one read. Here it is aligned at this position, and this same read here it is aligned at the same position, at this position. But here the read which is aligned, it is showing you the alignment percentage 90, and the same read which is aligned here at this position, it is showing you alignment percentage 20. So which will be more accurate position? Obviously 90%, right? So this thing we are getting here in this mapping quality, that how the read is aligned. All right, because let's consider in a DNA sequencing, if you wanted to call the variations, right? So you are checking the each and every read by aligning to the reference genome, and you are checking the base position. But if the read is not aligned at a proper position, and you are saying this is the variation or this is a mutation, your analysis will be wrong, right? That is why in the terms here, in the terms of DNA sequencing, this mapping quality is very important. But yeah, if you are doing the RNA sequencing at that time, we don't have to worry about the mapping quality because there we are not checking the base-wise alignment. There we are checking the read is mapping, how the read is mapping there. So that's it about the mapping quality. Now, the next and the most important concept regarding to the NGS is a depth and the coverage, which is combiningly called as depth of coverage. So what it is now? So basically depth of coverage or a depth, let's talk about first. It is telling like how many times the particular base or you can say a particular region is sequenced by the machine. Means here if you will see this diagram, let's consider this is what your target region. So how many times this particular target region or your region of interest or your base is sequenced by the machine? So here if you will see this particular region which is highlighted with the gray in color, here this particular region is read by the machine 5 times. Means here 5 reads are or 5 fragments are aligning. So at this region the depth is 5x. Now here this region if you will see, maybe I will show you by highlighting it. So here if you will see this region, at this region 5 reads are aligning. And in all those 5 reads, same base is present, so the depth is 5x. So how many times a particular base is sequenced? 5 times by the machine. So that is why depth is 5x. Now let's talk about this particular region. So this particular region is sequenced 2 times because here the 2 fragments are aligned. So that's why this particular depth is 2x only for this region. Now here, if you are talking about this region here, this particular region is read by the machine only 1 time. Means here only 1 read is aligned, so the depth is 1x. So if you will see here, we need a higher depth because higher the depth, it will increase the confidence that the particular region which is sequenced by the machine is accurate or it is correct. So that is why higher the depth, more the confidence. So you have to give a maximum depth whatever you want for this sequencers. And after the depth, let's talk about the coverage. So basically coverage means what? Like how many reads are able to cover the reference. So here if you will see this DNA, here it is covered by these reads. So this is what we call it as a coverage. Means if I am going to talk about one example, let's consider there is a 100 base pair long genome we are having and reads are only able to cover the 900 base pairs. So 100 base pairs are not covered. So at that time, coverage is what? 90%. Means if you have a 1000 base pair genome and only reads are able to cover only 900 base pairs. So 100 base pairs are not covered by the read. So the coverage is what? 90%. So also like we need a higher coverage. We need a high coverage. But when your coverage will increase, when the reads are able to cover more the reference, so when depth will increase, because when the depth will increase, you will get more and more number of reads, and when you will get more number of reads, it will able to cover your more reference. So which means like the depth and coverage are directly proportional to each other. So when your depth will increase, coverage will automatically increase. Okay, so this is what about the depth, and this depth you have to decide. You have to tell to the company that this much depth you want. But yeah, one more thing, higher the depth, higher the cost. Okay, so it's totally up to you that how much depth you have to give. So this is about the depth and coverage. So after the depth and coverage, the next term we are going to discuss, that is the file formats in the NGS. So there are different file formats we are having. So the most important or one of the important file format is a BAM and SAM. Actually, this BAM and SAM, these are the alignment file format. So basically here, whenever we are aligning our reads to the reference genome, so that output we are getting either in a BAM format or in a SAM format, depend upon which tool we are using. So BAM format basically it stands for binary alignment map format, which is in a 0101 format, which is in a computer readable format. So whatever the information is present here in the alignment, we are not able to understand in the BAM format. And another file we are having, that is a SAM format, which is a sequence alignment map format. And this file is actually human readable. Means both files contain a same information of the alignment, but one is in a binary format and one is in a human readable format. Because whenever we are performing the pipeline, so tools will be only able to accept the input in a BAM format, binary format. And also, like in case of large data, it is helpful to reduce the space also. And as it is a computer readable tool or program will be able to read that file correctly or properly or in a faster way. So whenever we have to give an input to any one of the tool, you have to give it into the BAM format. But maybe if you wanted to see how each and every file is aligned, how each and every read is aligned, so at that time you have to see SAM format. So basically SAM file format, it will give you the information in 11 mandatory columns. So what columns and how information we are getting, if you will see this figure, so it is started with at the rate symbol. So first of all here you are getting the header information. Okay, so here you are getting here the header information. Then here you are getting the sequence information. After that, here you are getting the actual information of these reads. That is your alignment section. And this alignment section, it will give you the information in 11 columns, 11 mandatory columns. So the description of each and every column is provided here, but even if I will explain you right now, you will not get that thing because we didn't perform any alignment yet. So whenever we are getting or we will get this SAM file in a real time uh, while performing the pipeline, that time I will explain you each and every column very detailedly. But for now, you just need to be understand that these are the output format of the alignment. So tool will only accept the input in a BAM, but if you wanted to see, you have to convert it into the SAM. And these files are easily convertible into each other. Like you can convert BAM into SAM and SAM into BAM using the tool called SAM tools. So what is the tool name? So SAM tools is a tool which is converting BAM into SAM and SAM into BAM. So how the SAM file is giving the information in those 11 columns, that we'll see during the pipeline. So this is what basically the alignment file format. Now the next file format we are having, that is a BCF and VCF format. So what is BCF and VCF now? So actually here, whenever we are performing the DNA-seq, we are calling the variation. So that variation information you are getting in this either BCF or VCF, means these are the variation calling file formats. So again, BCF, that stands for binary call format, which is in a binary language, computer readable language. So whenever you are getting the BCF file, you must need to convert it into the VCF file, because if you wanted to get the information of VCF variance, you have to read the VCF file only. So during the DNS-seq, our ultimate goal, our ultimate aim is to get this VCF file. That is a variant call format. So actually this VCF file is divided into three sections. So one is a metadata. Then next we are having that is a header lines. And then you're having that is data lines. So metadata actually it will give you the description about that file. Header lines will give you the actual headers and the data lines would give you the data of variants. But again, how we are getting the information and all that we'll see during the pipeline, because we don't have any VCF file, and even if we have, we need to be understand some concept before that. So that we'll see whenever we are getting the VCF file. But for now, here if you will see this image, so here in the VCF file you can see this is you are having that is a metadata line, means your first section. Then here you are having that is your header line, and then from this you are having your data line. So this is how the VCF file is divided into a three section. And the BCF file, if you will see, you are having the information in 0101 format, which you will not able to understand. So that is why we have to always convert the BCF file into a VCF file. So this is about the variant callings. But now, the most important file format which we have to discuss is a FASTQ file format. Because up to now we are only talking about the reads and reads. But in which format we are getting these reads? Whatever the data we are getting from the sequencer, in which format we are getting, that we are going to discuss now. So this raw data we are getting from the sequencer in a FASTQ file format. So just like we are having a sequences in a database which are submitted in a .fasta format. So likewise, the raw data, the fragmented data which we are getting from the machine, it's in a FASTQ format. So it's a raw data format. And this raw data format, it is a 4-line format. Means what? 4 line is equals to 1 read. Means let us consider if you're having 40,000 lines in your file, so 40,000 divided by 4, which means you'll have total 10,000 reads in your file, right? But as we are talking about the 4 lines, so what information we are having in this 4 lines, that we'll see. So the first line here. So basically the first line, it is always started with this @ symbol. And then it contains ID of your read. So the first line is always started with the @ symbol, which is unique identifier, and then read ID. Just like we are having a FASTA sequence, always started with the greater than symbol. So that greater than symbol is identifier of the FASTA, and the FASTQ identifier is a @ symbol. Then in the second line, you are having the actual read sequence, the sequence of your read. Then the third line, so basically third line you are having this plus symbol. So this plus symbol is indicating as a separator line. So this plus symbol is separating the second and fourth line here. But what information is present in this fourth line? Here in the fourth line you are having this ASCII character. Okay. So ASCII characters which represents the Fred quality score. As I told you in the Fred quality score term that we are getting along with the reads only, but in which format we are getting? In the ASCII character format. Means ASCII character, it is some combination of alphabets or a single digit number. So this is how basically we are getting the read sequences or a raw data. But one question can arise over here that why we are getting the ASCII character? Means machine is converting the Fred score into ASCII, but why? Why we are not getting directly the Fred score number? So actually it's due to the size issue, because if you will see about the number size is huge as compared to the characters. So that is why machine is converting the Fred score into ASCII character so that it will reduce the size. And the process of conversion of Fred score into ASCII character, that is that process we call it as a encoding. And this encoding is basically depend upon machine to machine. What I mean by is, let's consider this is what a symbol we are having over here, like this F symbol, let's consider for Illumina. So this F symbol is this F character is representing 20 Fred quality score for Illumina. But for Iron Torrent, 20 Fred quality score is representing some another symbol other than F. So that's what I mean is like here this encoding is depend upon machine to machine. So how and why it is like that, to understand this is one diagram here I have given. So if you will see this diagram carefully, here we are having the different sequences which are highlighted with the different colors. So let's talking about the Sanger sequencing, which is highlighted here with the purple in color, showing you the Fred quality score ranging from 0 to 40. Actually, whatever the example they have given over here is totally the hypothetical example. So you don't, this is just for your understanding. So here we are having a Sanger sequencing which is purple color and the Fred quality score ranging from 0 to 40. Now let's talk about the Illumina, which is highlighted with the blue in color. It is also showing you the Fred quality score ranging from 0 to 40, right? But here it is having the Fred plus 33 and here it is having Fred plus a 64. Actually, what happens inside the machine? What is this 33 and 64? That I will show you here. So let's talk about the Sanger sequencing. Consider just an example. So Sanger sequencing is representing maybe the AND symbol for 0 Fred quality score. And Illumina may be representing question mark as a ASCII character symbol for 0 Fred quality score. So why it is like that? Because machine is not able to convert directly the Fred score into ASCII character. So first of all, machine is converting Fred score into ASCII number, that technically we call it as a ASCII base, and that base is always start either with the 33 or with the 64, depend upon machine to machine. And then this ASCII base, like either the 33 or the 64, it is converted into the character. Now let us consider here if your Fred quality score is 1, 1. So here this will become 34. This will become 65. Now here it will represent some another symbol and here it represents some another symbol. So this is how actually machine is converting the Fred score into ASCII score, ASCII character by considering this ASCII base, which is either the 33 or with the 64. Now, if you will again talk about this diagram, so here you can see the Sanger sequencing, which is having the zero Fred quality score highlighted with the purple color. It is showing you 33 is a base and exclamation mark as a symbol. Now here if you will see the 40 Fred quality score, 73 is a base because 40 plus 33 is a 73 and here you are getting I as a symbol. Now here for Illumina zero Fred quality score 64 is a base and at the rate is a symbol. Means though the Fred quality score is same 00, but due to the different ASCII bases you are getting the different character and that's what the encoding means. So here basically you don't need to check it for each and every base like what Fred quality score represent what ASCII character because practically it is not possible. If you wanted to check the quality, as earlier I have explained the pipeline, we are checking the quality with the help of tool. So that tool will give you a graph and by seeing the graph we can quickly decide whether our data is having a good base quality or not. So you don't need to be worried about it. But still, if you wanted to see what base is representing what is like what Fred score is representing what character, ASCII character. So at that time here you can just type like Fred to ASCII and you can see this table. You can see any one of the table here where they are clearly representing. You can see Fred quality score, ASCII base 33 for the Illumina, Iron Torrent, PacBio and Sanger. So zero Fred quality score. Then this is the probability of error, the ASCII base and the character. And here you are having the sequencer which is having the base 64. You can see 00 Fred quality score both is having, but due to the 33 base it is showing you exclamation mark and due to the 64 base it is showing you the at the rate symbol as the ASCII character. So this is how you can check, but no need to do it manually. So these are the some basic concepts regarding to the NGS. Like whatever application you are going to learn, this concept must be remember this concept must be known to you. Then and then only you will be able to understand the NGS part. So that's it for today. Uh, in the next session, we are going to discuss how to download a raw data for from publicly available data sets and how to start the analysis. Till then, bye. Have a great day.